Our next panel is going to be about healthcare. So it is my great pleasure to ask Ishita Sharan to join us on stage. Ishita is a board member at the IIT Alumni Association of the Bay Area. She has been an active contributor to the community for several years. She is currently the general manager of healthcare at Konfer. She is a product of IIT Kharagpur, University of Southern California, and the Chicago Booth School of Business. Enthusiastic alumni, good. Ishita just organized a very, very successful women's leadership event last night, and she still has the energy to talk to us about advances in healthcare. So welcome, Ishita, on stage. Thank you.
Thank you, Swapnil. Good afternoon, everyone. Welcome to the panel topic today, Decoding the Multi-Omics Blueprint for Personalized Medicine, Strategy, Science, and AI. I know it's a mouthful, but it's also a topic that touches us all. Cancer and rare diseases touch all families. But it's not just the responsibility of doctors to tackle this disease. We need engineers, innovators, scientists, AI experts, all to come together. Multi-omics, in case the audience, if you're not familiar with the word multi-omics, it's simply looking at different aspects of biology, whether it's genes, proteins, chemical signals, and combining them to see stepping back and seeing the big picture of disease and health. But that's just the first step. The challenge is getting the science to a solution that's affordable, scalable, timely, and trusted. I would now like to invite on my panelists to the stage. So this is a very diverse group. We have the CEO of a large biotech company, a professor specializing in genomics models and framework, and CEOs of 2 startups, one of whom has a cloud biolab, and another a diagnostic tissue imaging platform. I feel this mix will make for a very rich discussion. Our discussion today will touch on 4 main areas. The first one is, I think it's okay. It's fine. But thank you so much for trying to follow my instructions. I instructed them when I was, while we were all waiting. So our discussion will touch on 4 main areas. The first one is describing the journey and what challenges they encountered. The second one is how each of them is advancing global innovation and collaboration and what regulatory hurdles they have to navigate. The third one, what opportunities technologists and entrepreneurs who are not in healthcare have, and if automation and agentic AI can help accelerate things. And fourth, if we have the time, we'll also cover what are the innovation gaps and where new ideas are needed. And finally, we'll wrap up with audience Q&A. So my first question is for Neil. So BridgeBio has been at the forefront of turning genetic knowledge into rare diseases and cancer therapies. I would like to invite you to introduce yourself and talk about challenges you encountered in moving from discovery to a drug that reaches patients.
Sure. Well, first of all, thank you, everyone, for having me, and I look forward to learning from the panel. Just by way of background, BridgeBio Pharma is a company that focuses in the area of genetic disease, as you mentioned, which for us encompasses both Mendelian or inherited diseases as well as cancers with clear genetic drivers. And that affects about 30 to 35 million Americans and hundreds of millions of people across the world. And when it does affect them, it affects with devastating consequence, being the 2nd largest driver of childhood death and morbidity. And the opportunity in this space really is multi-omic. For the engineers in the room, of which I think there are many, it's perhaps the only therapeutic regime that allows one to quantitatively describe the problem, going from genetic dysfunction to protein dysfunction to what's happening inside a cell, to what's happening inside a tissue, and ultimately how that presents phenotypically within the human, and then to target the causal driver at its source. And so retrospectively, that leads to much higher probabilities of technical success. It's not quite engineering success rates. It's still below 50%, but it's about fourfold higher than traditional drug discovery and development. So the opportunity is there thanks to the sequencing of the human genome by and large. The challenges that arise, though, are multifold. I don't have time to go through all of them, but I would say, first and foremost, the marketplaces associated with these genetic diseases are sometimes large enough for pharma to care about, sometimes they are not, and oftentimes the investment criteria for a biotech is, hey, I want pharma to be able to buy it or partner with me at some point. So one has to think carefully about the economic model. Second is, it still is a mode negative issue, so you're still mostly failing, and so how you fail and fail quickly without spending what can be hundreds of millions of dollars on any given program is an important thing to look at. And then finally, and these are all related, how does one efficiently move these drug products forward? Because we cannot afford to spend $2 billion per drug program. And this is where a lot of the new technology that's been talked about today, I think, is going to revolutionize, not so much new target identification and things of that nature. That will come, but I think in the near term, we have the ability to make drug discovery dramatically more efficient through fairly rudimentary applications of AI. So those are the types of things that, the challenges that have arisen for us.
Thank you so much. My next question is for Professor Kundaje. So cancer and rare diseases, they're also your interest, areas of interest. Can you please share your background with us and talk about what kind of open tools you create, how other scientists use them, and what engineering hurdles do you encounter?
Yeah, so just want to start by thanking the organizers for having me here. I'm an associate professor in the departments of genetics and computer science at Stanford. My lab is a computational biology lab. And as Ishita mentioned, we develop machine learning methods to try to decode the function of biological sequences. We try to understand the machinery inside cells that gives rise to their diverse functions and dysfunction. And we try to use these models to also study the genetic basis of diseases like cancer and various rare diseases. Our focus has really been on solving a fundamental challenge in biology, AI for biology, which is that we're usually working in a low data regime. So where AI has been very successful in other domains is where you essentially have access to massive amounts of data. And while there is the sense that biology also generates a lot of data, it's actually very sparse in specific zones. And so you often cannot build very large models just by scaling model architectures and so forth. So you've got to get very clever about how to introduce inductive biases into your models, build models that actually give rise to not just predictions, but also causal mechanism, because that's really the core interest. So we've developed a lot of open tools for model interpretation. Many of these tools are generalizable and can be applied to other domains, but we really specialize these for the genetic applications we're interested in. We build a class of models called sequence-to-function models. What these models do is they take biological sequences and map them to all kinds of multi-omic data. And once we train these models, we train them in a way that they learn causality so that we can do counterfactual perturbations. For example, you can take a genetic mutation, push it through the model, and the model predicts what kind of omic readouts would be disrupted in what types of cells. So that can help you do really exciting things like screening millions of mutations. We can deal with genetic variants that are common in the population, which affect things like Alzheimer's and cancer, but we can also use it to study rare diseases, genetic mutations that affect a single individual, so really personalized medication. I think one of the biggest challenges we face is actually getting access to data in the right biological systems. If you don't have access to the right data, it doesn't matter how awesome your model is, it's not going to work out. So that's one of the challenges I think we're grappling with, alongside improving models, making them causal and interpretable.
Awesome, thank you. My next question is for you, Samant. So sorry, we're sitting in the wrong order. I'll ask my next question to you, Rajat. So imaging is emerging as a new omics, as PathomIQ shows. In one of your videos I watched, you said that genomic tests have only 60 to 70% response rate. So please tell us about your work, the work that you are leading, and what challenges did you face building AI models that extract rich features for much better prediction and prognosis?
Thank you again to the organization and to give me the opportunity to talk today. So I'm Rajat Roy and CEO, co-founder of PathomIQ. PathomIQ's mission has been to address a long-standing need for very reliable predictive tests and accurate predictive tests so that the physicians in the clinic can prescribe personalized medicine for each cancer patient for a better clinical outcome with less toxicity. It's very important. Now, this is basically to advance the precision medicine for improved cancer care. Now, so to do that, what we have done is we have developed an interpretable AI platform that can actually use cancer patients' diagnostic tissue images, which are readily available during cancer diagnosis, and extract features to predict treatment response for that particular patient. And interestingly, as you know, that most of the genomic data are represented in units of RNA expressions. Similarly, we could take the whole slide images of a cancer diagnostic tissue images and extract units of phenotypes / morphometric features that can be mapped to patient outcome. Now, interestingly, the problem here is, as I said, is the lack of predictive tests. So what happens is that, in the clinic, the problem is that the cancer is a very heterogeneous disease. So that basically means that not every patient will respond to the same treatment the same way. And the physician in the clinic have no idea whether the patient will respond to the treatment, or even if they respond, whether they'll refract or have recurrence in 6, 9 months or 5 years. And based on that, the treatment intensification can be done. Now, there are a lot of genomic tests that you're referring to, which I mentioned, are there, which happens to be the ones which are being used by the physician in the clinic, but they have limitations. And one of the limitations is the accuracy of the prediction is not very high. The other one is they're exorbitantly expensive, and a lot of countries where it's needed in big numbers are not able to afford it. And more importantly, they require to ship the tissues to their labs and require sometimes 4 to 6 weeks to give the results. So we have been able to develop this platform that can give the answer without shipping any tissue, but just the images, and the answer in 2 hours with a prediction accuracy close to 90%. So it's kind of to tell you the truth, it's kind of a game changer in the industry, because now most of the people who could not afford it or could not get that kind of prediction accuracy are able to get that. And we have developed this test for prostate cancer, breast cancer, lung cancer, bladder cancer. Now, the challenge that you're talking about is that, it's very easy to get a lot of diagnostic tissue images. A lot of it is available. And most of the companies, what they're doing is they're throwing the data at the model, right? Now, the problem is when you do not have enough data, just like you mentioned, it's not data enough, especially when you're talking about prediction of treatment, you're talking about longitudinal data and outcome data. And for every indication, it's very, very scarce. So how do you do that? So in that case, you have to develop a model that is specifically tuned for that small amount of data, and that's the secret sauce for us that we were able to develop. For those small amount of data, how do we do that and still able to get such high accuracy? Otherwise, the notion of the AI is that they can tell you something, but they cannot say why they arrived at that. So we have been very careful from day one to make it interpretable. And the interesting part is not only we are identifying signatures from the tissue images, we are able to predict genomic mutations and RNA expressions from an H&E tissue. You don't have to go through a whole genome sequencing of the whole tissue, but we identify regions of interest from there. We are able to do that.
Thank you. My next question is for you, Samant. So infrastructure is at the backbone of innovation, yet often invisible. Can you please introduce yourself and share with us the challenges in building a cloud biolab platform that can support volume and variety of multi-omics data?
Thank you, Ishita, for inviting me, and thank you for being here. And it's interesting to hear just last talk what we heard, trust. You go to doctor, you go take a medicine, and all things, the trust. How do you build the trust? So I was in technology. I ran telecom, so many things, and we have to Six Sigma quality. You go through all the steps, and you have to make sure that it works fine. You debug everything. 2018, that's how I met Neil in some conference, and it triggered me that I think it should be solved because everybody has some personal thing. I have a LASIK surgery, and I think it's not ready for still, but people keep doing that. What happened that the laser puts there, it burns your cornea, and it creates imperfection. You have a scattering, 6 months, 1 year, it will not go away. You have low light performance. How are you going to handle that? FDA has approved it. It's going there, but there is no trust data there. How do you get it? And that's why when you get the drug discovery done, approved, after that also you need to keep feeding the data back to the system, FDA, so that look into that. So 2018, 19, I got few people, PhD, Stanford, all the places, to experiment on the mRNA because I have some personal story where I wanted to solve the cancer. When you start from ignorance, you can talk big ideas. Isn't it? So I thought, okay, I'm going to solve it. And then we started going slow progress, labs they were doing, and then COVID hit. You can't go to lab, but still you want to do it. Then it occurred to me, how can we run it remotely? And then I came back, progress was slow, manually you do all these things. I said, it has to be solved. We are doing pipetting. Life cells doesn't grow like in pipetting. It goes in some microfluidics or some tissue or somewhere. You need to provide some mechanism where it can be solved like that. So that's how it said, okay, microscope is also an instrument, isn't it? Without that, you can't do anything. So without instrument, biology can't be done. So we need the debugger, tracer, and all these things to make sure that whatever you're doing, is it traceable or not? Can somebody look into that one so that when we put, okay, FDA is one snapshot, they take it or run 10,000, 20,000, but what happens after there are millions of people there, isn't it? So you need to have something which can be seen, which can be looked into that. Cloud computing, blockchain, if you talk all these things, technology are there, automation is there, microfluidics is there, nanofluidics is coming up. So there are so many things there which you can do it. A lab scientist has challenging things to solve it, which they're running some protocols, hundreds of 200 steps. That's what multi-omics comes into that. When you add multi-omics, which is nothing but a snapshot of a system biology, how the system is behaving, how do you do that? If you have to have a whole time-stamp series data, think of it. And that is proteomics. It starts from genomics, epigenomics, transcriptomics, epitranscriptomics, just proteomics, post-translational modification, metabolomics. You just think about all the way it goes to your system, what is behaving, how it is behaving. It's a dynamic system. How do you do that? You can take one snapshot, but you can't do that. So we are at the cusp where we need to put all these things together and start whatever hypothesis we have in the system biology, we can verify that. Because multi-omics is your data to verify your system biology hypothesis, and didn't do that. So it's one and same, but a person can't run 1,000 steps. They will miss it. There was some single cell we were running that it was 78 pages of protocols were there, and you can do mistakes. You need automation. That's what it is. So multi-omics will be sold by instrument. People are going to put their hypothesis. You run yourself. It will run. It will provide you the traceable data. You can go and debug, like in the software we used to do the debug. Do we have microscope is one debugging method. Okay, you can look into that. You put label, without label. How do you handle it? There are so many things are coming into the picture, which we need to put it there. And then we all solve the system biology problem, which is going to be in the cancer. If it's not one, like I said, it's not a one-dimensional problem. Right. It's not gene to RNA to, it goes back and forth, and it's the whole feedback loop, the whole pathway get messed up. How do you figure out? The snapshots are not going to give you enough. Because one of the things happens in the endocrine system. You test in the morning, you get test in the evening, you test in the night. It's different. So how do you need to have tools and technology to help you collect all multi-omics data.
Thank you. Thank you, Samant. I would like to move on to the next topic, global innovation and collaboration. So question for you, Neil. How do you globalize innovation? And what are the biggest regulatory hurdles you face bringing precision therapy to global markets? And how do you navigate them?
Yeah, great question. I think it's very challenging to take new therapeutic agents globally. It's easy to get them obviously into the States and Europe, and it's becoming more and more challenging with every year. I would say it starts with the lack of regulatory harmonization across the world. I'll give you an example. We have an approved product that's now in 70 countries through either not-for-profit donations or in its commercial setting. And for each one of those submissions, it's a separate submission. Now I think we can automate some of those things, but there's oftentimes manufacturing regulations that are very bespoke based on the environment that's local. So that's one challenge that I think is huge, especially for smaller companies like ours. The second is pricing. Very obviously, there's some marketplaces that can command and do take on much more of the profitability of the biopharmaceutical industry than others. And obviously, with the current administration's take on most favored nation pricing, it becomes difficult to understand how one should commercialize globally to China, to India, to these types of areas. Thus far, we've done it through not-for-profit arms. Most of what we have are small molecules, so relatively low cost, and we give them away. On the other hand, that's not possible for gene therapies and some of the more expensive modalities that come about. But I would say the heartening part of this is that innovation is much more global now. Obviously, everyone's been reading about what's happening in China, but it is really truly happening in great academic universities worldwide. And so that aspect of drug discovery and development, I think, is becoming much more global. Availability of cutting-edge therapies, though, I fear will continue to be a challenge unless they're given away in a not-for-profit manner like we do.
Awesome, thank you. Question for you, Professor Kundaje, as well. So given your experience with ENCODE and roadmap epigenomics, how do you see collaborative projects evolve in the next 10 years? And do you see any data sharing challenges as you work across, different countries as you work with?
Sure. So just to orient folks, I was very fortunate to be part of two very large consortia. One is called the Encyclopedia of DNA Elements. The other was called Roadmap Epigenomics Project. These consortia started right after the Human Genome Project. So Human Genome Project, we obtained the first draft sequence of the human genome. These other projects try to get these draft regulomes. These are molecular multi-omic maps of hundreds and thousands of cell types and tissues, sort of a reference map for all of humanity. And these projects took about 20 years. They were largely academic collaborations. They have been quite successful and generated the first wave of data that were used to train some of the latest and greatest AI models in the space. So looking forward, I think there are many interesting axes which I think could improve some of these large-scale efforts. The first is traditionally what has happened even in these large projects is the data generation efforts are initially fulfilled, a lot of effort and money is put into those, and the modeling aspects come later. Now, when that happens, often what ends up happening is you end up with large amounts of data, which may or may not be ideal for modeling purposes, right? And there's really a chance now to kind of make the feedback loop a little bit more flexible, where AI models come in at the very beginning and help with experimental design, thinking about what exact data sets you need to collect to push models to the next level. I think AI has really kicked in many ways in biology, and so now is a good time to think about how models can play a part before data generation efforts. I think the other aspect is of technology development itself. So often there is an impetus to generate a lot of data with the technology we currently have. But sometimes the data you really need to push the field forward is not yet there because the exact assays, the experimental platforms are required, need to be invented yet. And so, trading off, ENCODE in its early phases had a huge tech dev component. For example, RNA sequencing, which is like very common now, was invented in that time, right, through an academic consortium. The focus now is much more on data generation and scale. I think there should be more effort also on the technology development side of things to push boundaries on zones of multi-omic measurements that we simply cannot collect at scale highly accurately. And the third is industry-academia partnerships. So a lot of these consortia in the early days were either purely academic or in fact contests between academia and industry. I'm sure you know the story of the Human Genome Project and how there was a time when academic opportunities are lagging behind industry, and then there was a race. I think we're in a better situation now, and it's really, I think, an interesting time for thinking about how academia and industry could bring the best of both and push all of these efforts to the next level. So that's where I hope things will go in the future.
Awesome. Thank you. Question for you, Rajat. You have already partnered with Newberg Diagnostics, deploying urea biomarker tests internationally. What challenges did you face with regulations, and did that shape your strategy in any way?
Yeah, so thank you for that question. So I'll step back a little bit and talk about our approach first in the United States a little bit, and then go into the global perspective. So in the United States, we are addressing two markets. One is working with the big pharmaceutical companies, as well as the clinic. So there, we took the approach of, from the clinical perspective, we wanted to take a lab-developed test approach where we can get the test out from the lab itself, from a CLIA-certified lab. Whereas while we are working with FDA, but FDA allows you to, go out and launch your products as lab-developed tests. Now, in the case of globally, we are actually addressing the market, Asia-Pacific market. We're working with Asan Medical in Korea, China Medical in Taiwan, and Singapore Hospital, and Tata, as well as All India Medical Institute. So what we took the approach is first to go directly to those hospitals and address their needs. So we had to go through a huge amount of validation and testing with their data. But having said that, each country has their own, Korea has their own FDA sort of challenges, Singapore has it all. So we are going through those challenges at this point to ensure that we can, not just for the hospitals, but we can deploy to the rest of the countries.
Awesome, thank you. So question for you, Samant. As you innovate in the bio-cloud lab space, how can, so as you try and expand to underserved communities, how do you work on making sure access, keeping access is compliant and equitable? Because we talked about this, that you're passionate about getting this to India.
Yeah, thank you, Ishita. That's an interesting question. So if you look into that, every regulatory agency does something, but still they look for the actual data, whatever is there. By building the cloud lab, by running it 10x cheaper, 10x faster, what happened that first of all, you reduce the cost. And second thing, you have the traceable data. And then the second aspect is going to be translating that into the regulatory regime of each country. So that's another layer you put on top of that. So that gets simplified because every regulatory agency, like in US also, if you have done some test in Europe or Australia or many developed countries, they are also coming into that one. They can use that basically if you have sold something. So there is a credibility issue comes into that. So that reduces even the cost of many of the clinical trial. I think you guys are going to run that we are not doing, but you guys are doing. Ultimately, that is going to reduce. So the total overall cost is going to get reduced. And that's the right. Plus the diversity. When you put the cloud lab, you have people coming from different places. Since one of the major change happens in the cloud lab is that you have a data there, you can run samples there from anywhere in the world, you see everything. So every regulatory agency can see the same data. Of course, you have to make sure that it's traceable, trustworthy, and all those things. And that's going to reduce and help those regulatory agencies to accept that one. So that's what from my technology point of view, I see that.
Thank you. So in the interest of time, we can want to quickly cover the next topic, which I think is of high interest to our audience here. So this is a two-part question. You can choose to answer either of those two parts that apply to you. What opportunities do you see for technologists and entrepreneurs who are not from healthcare? And do you see a way for automation and agentic AI? I know you touched on that. Agentic AI to accelerate progress in this field. It's about 30 seconds each, if you can, either part of those two questions. I'll start with you, Neil, if you have. Yeah.
Well, I'll go with the boring stuff. I think if you look at the economics of clinical trials, the largest loss happens in the interstitium between phase 2 and phase 3 and phase 3 and approval. And there's a lot of technology involved with database close, regulatory submission, much of the statistical analysis that's required, and even site audits. And all of that the technology is extremely archaic. So technologists who are looking at, I think, the biotechnology space could really innovate in that sector and provide huge savings to the industry and more efficient drug discovery and development so we can get drugs to patients more quickly. So I think that's a huge unmet need.
Okay. Professor.
Yeah, maybe I'll talk about the agentic component and sort of focus on one case study, which is rare disease diagnostics, right? So rare disease diagnosis is particularly challenging because, as the name suggests, rare diseases are diseases that affect like very few individuals. That means you don't have big references to go and look at. They're literally like, you're trying to solve a puzzle, which is really hard. Often requires, a combination of clinical geneticists, genetic counselors, biologists, folks who do AI, all coming together to solve a problem. You have to often put together everything ranging from models to databases to prior knowledge to similar cases. It's an excellent scenario for agentic systems to come in and really speed up the process. It's not like they're going to come in and solve the problem, but they're going to dramatically improve the bottlenecks that exist in the process. And for a lot of rare disease diagnostics, speed is of the essence. These are usually diseases affecting young children. They don't have much time. So the faster we get through it, the better it would be. And there's already lots of exciting work in the agentic space to speed up the workflows, to simply solve the problems faster.
Thank you. Question for you, Rajat.
Yeah, so to answer your first question.
Sure.
So I'm a prime example. I'm a computer scientist by training. So, having done 3 previous startups, this is an example that there was a problem that needed to be addressed and used our own background in the high technology to address that. So to answer your question, what can be done? There's a tremendous amount of problems that needs to be solved in this healthcare domain, and people should be using their know-hows, what they have developed to address, because there's tremendous amount of information. On agentic AI, as we talked about, you addressed that, people are building foundation models to extract features, then do what? Like you have to answer specific questions for each one of them, and that's exactly what we are trying to do, is there's a lot of information that you can gather, but how do you use that data to answer questions? And that's where the agentic AI is coming into play.
Thank you. Samant.
Yeah, so I think this is the right time for every technologist, electrical, computer, doesn't matter, to get into the life science, because life science is nothing but movement of molecules, atoms. That's all. It's just at different scale it goes. So everything applies what you do. It's a big data issues, big protocols. You need to do all these things. And I don't think there will be any better time than now to do anything in the life sciences, because it's for everybody, for your own thing. And agentic, when I'm talking about that, so agentic basically provides you, for us basically, it's like 100x improvement in any protocol. You write it, it can go tell you immediately. Of course, you need to put guardrails, figure out whether it is hallucinating or not hallucinating. That you have the whole action space, we call it, so that it cannot go beyond that. Like people are talking about co-scientists and all those things. So it starts all with the image analysis, do the whole segmentation and all those things, Multi-Omics, you were talking about. You go through every step, starting from all the way to the bottom in the chain to how you make the whole protocol, how do you verify what is happening, not happening. It's very simple when you have AI and deep learning also part of that, not just agentic. Agentic is one part, but you have. So think of it, you have hundreds of PhDs sitting there helping you out running protocol. It can't be better than that.
Yep. Awesome. Thank you all so much. And we are out of time, so I don't think we can take questions. Do we have time?
No,
Unfortunately,
please.
no.
Yeah, we don't have time. We're running late.
The panelists will be available after the session, so you can talk to them. So thank you all so much. It was wonderful learning about your background, challenges, how you're doing global innovation, and opportunities for the audience.
Thank you.
Yeah.
Awesome. Thank you, Rajat. Yeah.